So it is my honor and great pleasure on behalf of the organizers to introduce my colleague Larry Zipursky as today's keynote speaker. So like many of the greatest American scientists, Larry is originally an immigrant, Canadian, who came to the US for his scientific education. Larry was first a graduate student of Gerald Hurwitz at Einstein, where he studied bacteriophage DNA replication. And he then made an extraordinary transition as a postdoc to Seema Benz's lab at Caltech, so working on Drosophila, where he worked on developing antibodies as a critical resource in developmental biology. And I'm going to use Larry's own words here to describe what he did, or his insight, that monoclonal antibodies highlight pattern elements that are not readily observable by other techniques. Monoclonal antibodies can be used to identify the molecules associated with particular patterns, providing links between observable structures and the genes. So why is that remarkable? Because this was in the early 80s. And it is something that we completely take for granted now, but this insight, together with molecular cloning, in my opinion, ushered in the revolution in developmental biology that we are now still living in. Because you can't study a process unless you can identify it. And I'm sure all of us in this room will have an example of a favorite antibody that made the studies in our model system possible. I'm shouting out to tag one there. So Larry moved across town to UCLA to start his own lab in 1985, where we've been ever since. And I'd like to think that's because we're so collegial at UCLA, but it's also really hard to leave Southern California. He has won almost every prize you can think of, including becoming HHMI in 91, and being elected to the National Academy of Sciences in 2009. And I feel confident that there are more prizes to come because Larry has spectacularly capitalized on the extraordinary power of Drosophila genetics to make seminal contributions in our understandings of the mechanisms that specify neuronal connectivity. So just very brief. Larry started out using the fly eye as a model system for neural cell fate and identified critical regulators of the receptor tyrosine kinase family. And that gene is called Bridle-7less. And anybody wants to know more about that, find a faculty of at least my age, and we will tell you more, because we love to talk about that story. So he went on to identify DSCAM1 as a gene that can be spliced into a remarkable number of isoforms that then, with remarkable specificity, permit neurons to distinguish self from non-self. And unraveling that mechanism was a tour de force of molecular genetics. Most recently, he's broadened his focus to the mammalian visual system and continued working on the Drosophila visual system, setting his sights on identifying all of the synaptic specificity molecules through connectomic and bioinformatic approaches, and that's more tour de force work he'll be talking about today. So I just want to end on a personal note that I have been in awe of Larry since I was an undergraduate. I learned about the genetics of the fly eye, and I used that model system in my thesis work. And Larry won't remember this, but he came to my poster at the fly meeting when I was a graduate student, and it made the meeting for me. And I wanted to give that shout out to all the faculty, 2026. Go to student posters. They remember. I still do. And just finally, one very LA story, though. So when I first moved to LA, I was sure that I'd never see celebrities, and that's true. I never do. But a couple of months in, I actually did see somebody very famous leaving a well-known restaurant. And I gripped the arm of my companion and said, "Oh, that's Larry Zipursky." So please join me in welcoming Larry, who is talking on cell recognition molecules regulating the assembly of the Drosophila visual system.
Well, I can only disappoint. Thanks, Samantha, for that definitely over-the-top introduction. And I'd like to thank the organizers for the opportunity to speak at this meeting. It's really an honor and a privilege to be here. The first meeting in this series was in 1998, and it was really a landmark in the field. It marked the transition from what was previously largely a descriptive field to a molecular one. Talks focused on the enormous progress made in identifying guidance molecules and the receptors. There was, however, very little discussion about how synapses formed. It's been nice to see at this meeting, hear what progress has been made in understanding how synapses are formed and how they're modulated. What I want to focus on this afternoon is a really, really messy, poorly defined step between axon guidance and synaptogenesis. When axons and dendrites have reached the target region and have to select correct partners from many incorrect ones available to them. And I'm going to refer to this as synaptic specificity, which I recognize is somewhat of a loaded term. Here, intermingled axons and dendritic processes discriminate between an often staggering array of potential partners. The result of these interactions is a predictable choice between potential partners. Presynaptic neuron A chooses B over C. It can be an all or none phenomenon, or it can be a graded preference for synapses. How are these choices made? What are the developmental strategies underlying these choices, and what are the molecules that execute them? And to address these questions, my lab is focused largely on understanding this problem in the Drosophila visual system, and we've done so for several reasons. First, there's a dense and very dense synapse-level connectome of the fly visual system. There are powerful molecular and classical genetic tools that allow us to carry out gain and loss of function analysis. We have a comprehensive single-cell sequencing database for hundreds of neuron types as they're selecting their synaptic partners. We can follow this in real time, imaging specific neuron types in developing brains in intact animals. And finally, there's a rich repertoire of visually guided behaviors to assess the consequences of changes in synaptic specificity on animal behavior, and neural activity can be measured electrophysiologically as well as through calcium imaging. The fly visual system comprises the retina, that obviously detects images, and the optic lobe that processes visual information. And the optic lobe comprises four parts: the lamina, medulla, lobule, and lobular plate. There are four classes of neurons: optic lobe intrinsic neurons, which make their connections within a given region; optic lobe connecting neurons, which make connections between different regions of the optic lobe. There are visual projection neurons that extend their dendrites into the optic lobe and axons into the brain. And then conversely, there are neurons which extend their dendrites into the brain and their axons into the optic lobe. And there are many neurons, many types of neurons within each of these classes, and each of them elaborates distinct patterns of synaptic connectivity. Here I've summarized the visual system connectome from a recent paper, just very recent paper on bioarchives from work at Genelia. And in this paper, they report 53,000 visual system neurons that have been reconstructed. 727 neuron types have been defined largely morphologically and by their patterns of synaptic connectivity, and there are 49 million synapses. Certainly enough to make you think that this is the time to retire. Now, neurons form a range of synapses from thousands to less than 10. Most neurons are pre- and postsynaptic to multiple neuron types, and virtually all synapses in Drosophila are multiple contact synapses, meaning they have a single presynaptic release site that releases neurotransmitters onto multiple postsynaptic elements, rarely from the same cell, but typically from multiple different cells, different cell types. This is an EM section through the M3 layer of the medulla, and here the processes of two amacrine-like cells, DM12, form synapses selectively with this terminus of the L3 neuron. They do not form synapses with other neuron types. So how do these axons and dendrites discriminate between one another in this sort of spaghetti-like mix, selecting some neurites to make synapses with and to avoid others? RNA sequencing studies in the developing fly olfactory system from Leach and Lowe and from the visual system from my lab and Claude Desplan's have shown that there's a marked increase in the number of cell recognition molecules expressed in different neurons, even very closely related ones as synaptic circuitry is established. These recognition molecules fall into several families, including hundreds of genes, including immunoglobulin superfamily proteins, and a more modest number of leucine-rich repeat and cadherin-containing family members. In addition, guidance molecules such as semaphorins, efferents, slits, and netrins and their receptors are also expressed. Each neuron type expresses literally hundreds of these proteins in different combinations. So much of the cell surface molecular diversity in Drosophila, in the developing neural pill, is contributed by proteins of the immunoglobulin superfamily. In this heat map, I've summarized the expression of a subset of Ig superfamily proteins expressed during synapse formation in just 14 different cell types, and the cell types are on the right. Each row is a different neuron type, and each is divided into 7 rows, representing expression at 12-hour intervals during a protracted period of synaptogenesis during pupil development. There are 4 families of proteins, some of which you've heard about at this meeting: DIPs, DPRs, beats, and sides, and they're encoded by 54 different genes and form 2 interacting networks of recognition molecules. And I've only shown you a part of those interactomes on the right-hand side of the slide. The colored lines in the DIP-DPR interactome represent different affinities, ranging from the low micromolar up to over 100 micromolar. The first DPR was discovered by Craig Montel in a behavioral screen, and Ozgan and colleagues identified additional paralogs and identified their interaction proteins called DIPs for DPR interacting proteins. And a quantitative and detailed analysis of these interactions was reported by Kaminescu and colleagues in the Shapiro and Honig labs. Now, the founding members of beats and side families were identified the old-fashioned way in genetic screens by Corey Goodman's lab many years ago. Herman Aberle and his colleagues demonstrated that these proteins were a ligand-receptor pair. And then Engin Ozgen, Zinn, and Garcia then identified additional paralogs of beats and sides and defined their interactome. Now, these genes are expressed in complex cell type and temporally specific patterns, endowing each neuron type with a unique cell surface identity. Neurons typically express 1 or a few DIPs. They express a lot of DPRs, as you heard from Hagar's presentation yesterday. Lots and lots of DPRs are expressed in very complex patterns. And then the expression of beats and sides sort of falls somewhere in between the expression of DPRs and DIPs. Highly related neurons, such as T4A and T5A, which are on the bottom of this slide, have very similar patterns of synaptic connectivity, and they have very similar patterns of gene expression. By contrast, L1 and L2 and L3 lamina neurons that are at the top of this slide have a very common origin. They have very different patterns of synaptic connectivity, and they express very different combinations of these 4 family members. Now, regulation is not restricted to transcription. As seen on the left columns, DSCAM1 and DSCAM2 are expressed in all neurons. And as we showed many years ago, transcripts from these genes are alternatively spliced to give rise to isoforms with different recognition specificities. For DSCAM1, this gives rise to 18,024 extracellular domains, really quite a ridiculous number. And each of these exhibits isoform-specific homophilic recognition. And we know from genetic analysis that thousands of these are required for normal development. So this, I think, really underscores the need for massive molecular diversity in brain wiring. You heard a wonderful talk about this analogous family last night with the protocadherins. Now, though DSCAM2 is expressed in all neurons, MBL, which is the most left-hand side, is a cell type-specific splicing factor. and it regulates alternative splicing of DSCAM2 to give two different alternative isoforms, each of which shows isoform-specific homophilic recognition. Other post-transcriptional mechanisms also contribute to wiring. For instance, although turtle, also shown here on the right-hand side of the left-hand block, is expressed quite broadly, it's highly localized to specific subcellular domains in many cell types. Now, these proteins function in very different ways. So DIP-DPR and beat-side interactions have been shown to mediate synaptic partner matching in my lab as well as in the Zinn, Carrillo, Mann, and Schuldiner labs. In this example, DM12 expresses DIP-alpha, and L3, its sole synaptic partner in the M3 layer, expresses its interacting proteins DPR6 and DPR10. Loss of function of DIP-alpha leads to a 30% decrease in synapses in M3. You can't see that decrease up here. You'll have to take my word for it or read about it. And you can also see that it forms ectopic connections in a deeper layer, DM8 layer. Targeted expression of DPR10 to a deeper layer then leads to mistargeting of DM12 to this deeper layer in a DIP-alpha dependent way. Thus, these and other data argue that this interaction is both necessary and sufficient for targeting synapses between these neurons. Now, all synapses in Drosophila are multiple contact synapses. And the best characterized of these is a tetrad synapse between photoreceptor, presynaptic terminals, and postsynaptic elements from different kinds of lamina neurons. And a feature of this is that there's an invariant pairing of postsynaptic elements from L1 and L2 neurons, and DSCAM1 and DSCAM2 together play a crucial role in ensuring that pairing. L1 and L2 neurons express a unique combination of DSCAM1 and DSCAM2 isoforms. To remind you, both DSCAM2 and DSCAM1 promote homophilic repulsion in other contexts. For DSCAM1, that's to promote self-avoidance, and for DSCAM2 to promote tiling, axon tiling. So, as you can see, removal of DSCAM1 or DSCAM2 alone results in only very modest defects in the composition of the postsynaptic elements. However, removing both DSCAM1 and DSCAM2 together in mosaic animals randomizes the pattern. As postsynaptic elements from the same cell express the same DSCAM1 and DSCAM2, they recognize each other, they bind to each other, and as a consequence of recognizing and binding to each other, they dissociate from one another, are repelled, and that ensures that there's invariant pairing of L1 and L2, that you don't have any L1, L1, or L2, L2. L1 is in the on pathway, L2 is in the off pathway. It's unclear why you need to have this strict coupling, but it is literally invariant through thousands of examples that have been looked at. Thus, in this case, the correct match arises by a mechanism that prevents inappropriate pairing of postsynaptic elements of the same cell. And finally, synapses exhibit subcellular specificity. So in this example on the right, different neurons form synapses in specific subcellular domains along the proximal distal axis of T4 dendrites. MI4 is GABAergic, MI1 is cholinergic, and MI9 is glutamatergic. And this subcellular arrangement plays an important role in computing directional selectivity in these neurons. Now, specific features of this connectivity pattern led Piero Sanfilippo in the lab to speculate that cell recognition molecules associated with neurotransmitter receptors would determine the specificity of the synaptic input to different domains. To assess this, he first tagged neurotransmitter subunits by modifying the endogenous loci, encoding them to determine the distribution of these receptors in T4 dendrites. He demonstrated that each receptor subunit exhibits a distinct distribution. He then used the tag as an affinity reagent to purify recognition molecules associated with synapses and to determine their identity by mass spec. he discovered that each receptor was associated with a distinct set of cell recognition molecules. Turtle, for instance, a fly homologue of mammalian Ig superfamily protein IGSF9B, is localized to the proximal region of T4 dendrites, and it's required for the development of GABAergic synapses within this domain. By contrast, MMD is localized to the tips, the distal end of T4 dendrites, in its association with the glutamate receptor, gluci-alpha, and it's required for the formation of synapses, these synapses in the distal domain. Side family members, side 6 and side 7, were associated with different acetylcholine receptor subunits in the more central domain, and Piero's in the process of asking what role they play in synapses in these neurons. So Piero, on the left there, named, presented this in a poster yesterday, and I know the posters are down, but if you have a chance to you can now see him, recognize him. If you see him around, really pin him down and ask him to tell you about this. It's really a wonderful story and has lots of promise to tell us something new about synaptic specificity. Now, having a dense connectome and ways to interrogate the relationship between circuitry and behavior provides new and really exciting opportunities to identify new wiring strategies and then to explore the underlying molecular logic. And for the rest of the talk, I'd like to focus on Mark's unpublished work, which has provided insight into yet a different strategy by which Ig superfamily proteins contribute to synaptic specificity. And these studies really began when Mark and I had a conversation with Gwyneth Card, then at Genelia, now at Columbia, before the pandemic, and she told us about a discovery she had made. She works on the escape behavior in flies and the neural circuitry underlying it. And she had discovered a wiring motif, which was new to us, which converted positional information in the retina into different numbers of synapses at a sensory motor interface in the brain. And she encouraged Mark, who's recently a recently arrived postdoc in the lab, to explore whether such synaptic gradients were more prevalent in the connectome, and then to explore how genes controlled them. VPNs, or visual projection neurons, are feature-detecting neurons. There are many different types of VPNs. Their dendrites uniformly cover the visual field within the lobula or lobula plate. These neurons extract information, specific features from images that fall on the retina, and are processed by circuitry linking photoreceptors to VPN dendrites. Different VPNs respond to different stimuli, such as the movement of small objects or looming. The axons of VPNs converge onto discrete targets in the brain that transform this information into behavior, sometimes directly synapsing on dendrites of descending neurons, which regulate specific actions. This image shows sparse labeling of some of the 100 or so LPLC2 neurons and their dendritic arbors in the lobula and lobula plate, as well as their axons converging onto a small target in the brain referred to as the optic glomerulus and optic glomerulus. There are more than 20 distinct VPN types with dendrites that uniformly cover the visual field in the lobula and or lobula plate, which are tuned to different visual stimuli with axons that converge onto different target glomeruli in the brain. Today I'll focus on the synaptic inputs to LPLC2 dendrites, which are tuned selectively to respond to a looming stimulus, and the synaptic specificity of LPLC2 axons onto giant fiber dendrites, which drive the escape response. This is an example of the escape response in a single fly, responding to a small looming stimulus, and this is driven by LPLC2. LPLC2 shows an increased sensitivity to looming stimuli in the dorsal visual field, and you can see that in the histogram on the right. This dorsal selectivity correlates with two synaptic gradients. EM reconstruction revealed a dorsal ventral gradient of synaptic inputs to them from motion-sensitive T4, T5D neurons onto the dendrites of LPLC2 neurons in the lobula plate. T4, T5D neurons from different parts of the visual field convey information to dendrites of LPLC2 in a topographic fashion. So dorsal dendrites receive many more presynaptic inputs than ventral ones. LPLC2 axons form a second synaptic gradient with the dendrites of the giant fiber. Here, the axons of all LPLC2 neurons lose their topographic arrangement as they converge onto and intermingle with the dendrites of a single giant fiber neuron. Axons from dorsal LPLC2 neurons form more synapses onto the giant fiber than more ventral ones. So here we have retinotopic information in dendrites, which is transformed into gradients of synaptic inputs to a common target. We first set out to identify the molecular basis of the synaptic, and when I say we, it's Mark, who, by the way, will be on the job market. And so we is Mark, and I'll say we a lot because I don't want to keep saying Mark. So we first set out to identify the molecular basis of the synaptic gradient formed by LPLC2 axons onto giant fiber dendrites. So as axons lose their topographic arrangement, we reasoned that the simplest model to account for these gradients would be graded expression of cell recognition molecules in the LPLC2 neuron population. To search for these, we used a single-cell sequencing approach. Now, our single-cell atlas of the developing visual system put together by Yuval Kurmangaliev and others in the lab, we had sequence of only a very small number of LPLC2 neurons. With such a small population of neurons, variation in the number of transcripts within this population would not have been meaningful. So to increase the number of LPLC2 neurons sequenced, we labeled these cells with a transgenic marker, purified them by cell cytometry, and sequenced them. To explore heterogeneity in gene expression across the LPLC2 population, we carried out principal component analysis. The first principal component captured a significant group of genes that were expressed in a graded manner across individual neurons. Genes are in rows here and are ordered by their loadings to PC1, and each cell is in a separate column, and these are ordered from left to right by their PC1 scores. These gradients were verified using FISH on expanded tissue. The red arrow shows a strongly positive sialotransferase cell with low levels of DPR13. The blue arrow shows a cell with high levels of DPR13 and low levels of sialotransferase. The lower left inset shows the location of the LPLC2 cell bodies and between the brain and the optic lobe. And the inset in the upper right shows the graded expression of these two genes. GAL4 reporters then allowed us to determine the polarity of this difference in expression. DPR13 expressed high levels dorsally and low levels ventrally, and sialotransferase showed patterns of the opposite polarity. And today I'll focus on two of the genes that came out of this, DPR13 and BEAT6, both which have a dorsal to ventral gradient. Now, DPR13, BEAT6, and DPR17 are expressed in a graded fashion in this population of cells. So synaptic gradients could be driven by high DPR13 or high BEAT6 patterning through adhesive interactions with their interacting partners, DIP-epsilon or Cy2. Or alternatively, DPR17 could inhibit synapse formation in a graded way from ventral to dorsal through its interaction with DIP-gamma. We used FISH to assess whether these binding partners were expressed in giant fiber neurons. Here, the giant fiber neuron nucleus is marked with GFP. The giant fiber expressed high levels of DIP-epsilon. It did not express DIP-zeta, which is a paralog of DIP-epsilon that shows the same DPR binding specificity. It expressed very low levels of Cy2, which interacts with beat 6, and it did not express DIP-gamma. We next set out to assess whether DPR13 then and DIP-epsilon regulate synapses between LPLC2 axons and giant fiber dendrites. Removal of DIP-epsilon decreased the overlap between LPLC2 axons, shown here in red, and giant fiber dendrites in green, and this is quantified in the upper right. This was reversed by targeted expression of DIP-epsilon cDNA in giant fiber dendrites, and what I've not shown you here is that the null allele was phenocopied by targeted expression of 2 different RNAis to the giant fiber. DPR13 nulls regrettably do not have a phenotype. However, 4 other DPR paralogs are expressed in LPLC2 and bind to DIP-epsilon with similar affinities, and this is complicated loss of function genetic analysis. Oops. So to mimic the effect of loss of function of these 5 DIP-epsilon interacting paralogs, we ectopically express DIP-epsilon in LPLC2 to, at least in principle, sequester DPRs in cis complexes and thus prevent interactions in trans with DIP-epsilon on the giant fiber. This ectopic expression in LPLC2 phenocopies the loss of function of DIP-epsilon in the giant fiber. So together, these data show that DIP-epsilon and DPR13 and one or more of its paralogs regulate connectivity between LPLC2 and the giant fiber. So we next sought to assess whether changing the graded expression of DPR13 altered the synaptic gradient. So removing DIP-epsilon from the giant fiber through targeted expression of DIP-epsilon RNAi resulted in reduction in the giant fiber response to LUMI at all elevations. By contrast, overexpressing DPR13 eliminated the dorsal selectivity of the response, increasing the response at 0 and 45 degrees without changing the maximum response in the most dorsal region. And that's shown in this last panel. In summary, then, these data are consistent with a gradient of DPR13 in LPLC2 axons, leading to a gradient of synapses onto giant fiber dendrites through its interactions with DIP-epsilon. So what controls the gradient of synapses of T4, T5D onto LPLC2 dendrites? Here, T4, T5D neurons from different parts of the visual field form synapses to topographically matched LPLC2 dendrites in the lobular plate. The number of synapses on dorsal LPLC2 dendrites falls off to very low levels ventrally. So the left-hand panel shows you a single LPLC2 neuron in red. The dendrites extend into the lobula and then the lobular plate to the left. T4, T5D axons form synapses onto the dendrites of LPLC2 in the lobular plate. The upper middle panel is a false color image of dendrites of a single LPLC2 neuron. It's an incredibly cool neuron. Each LPLC2 neuron has 4 dendritic branches, one in each lobular plate layer extending in one of the 4 cardinal directions. The colored triangles to the left represent the preferred directional response of dendrites in each layer to motion stimuli. Branches respond optimally to motion from the center of the receptive field outwards. So the output of LPLC2 axons is a nonlinear sum of the activity of each of these 4 branches, such that individual LPLC2 neurons serve as small object looming detectors. T4, T5D form synapses selectively on the branches in layer 4, as shown in the bottom in the middle panel, with A, B, and C types of T4, T5 neurons forming synapses selectively in branches in layer 1, 2, and 3 branches, respectively. In addition to selectively forming synapses in branches in layer 4, EM reconstructions revealed that T4, T5D form a gradient of synapses along the dorsal ventral axis, with many more synapses on dorsal LPLC2 than ventral ones. So here, on the right of the gradient, shown on the right, is we visualize by color coding each of these LPLC2 neurons by the number of synapses T5D neurons make onto the branch of LPLC2, of each LPLC2 in layer 4. So these findings suggest that dorsal and ventral LPLC2 neurons within one animal receive differential amounts of upward and downward motion information. So what's the molecular basis of this gradient? So to remind you, like DPR13, BEAT6 was also expressed in a graded fashion in LPLC2 by single-cell sequencing, and this was also verified by FISH. As shown here, using BEAT6-GAL4 reporter constructs, this gradient of expression is also along the dorsal ventral axis with high dorsal expression and low ventral expression. Now, in a previous study, we showed that BEAT6 binding protein SIDE2, so this ligand receptor pair, is selectively expressed in T4, T5D. Furthermore, in this earlier paper, we showed that SIDE2 is necessary for T4, T5D connections to other BEAT6 expressing neurons with dendrites in layer 4. So these observations led us to test whether BEAT6 SIDE2 interactions also regulate the graded connectivity to LPLC2 branches in layer 4. Targeted expression of BEAT6 RNAi led to a graded phenotype with loss of the branch in layer 4 in the dorsal lobular plate, a reduction in arbors in the central region, and it had no effect on ventral neurons. And these data are quantified on the right. BEAT6 nulls, as well as loss of SIDE2 in T4, T5D neurons, also led to a similar graded phenotype along this axis. We next sought to assess whether these changes in LPLC2 branching morphology affected the tuning properties of LPLC2 using calcium imaging of tethered flies in flight. Flies were presented with a series of very small objects moving in different directions. In control flies and in flies with BEAT6, where BEAT6 was selectively removed from LPLC2 using RNAi. Using 2-photon imaging, Giovanni Frighetto and Mark Fry's lab at UCLA imaged LPLC2 neurites, in a segment between the branches in the lobular plate and lobula at different dorsal ventral positions. As shown on the right, targeted expression of RNAi in LPLC2 neurons altered the directional selectivity of the response, with a decrease in the downward response of dorsal LPLC2 neurons. So this is consistent with the morphological defects which I showed you on the previous slide for both BEAT6 and SIDE2 mutants. So in summary, 2 gradients of cell recognition molecules shape synaptic gradients in different neuronal compartments of the same cell. More beat6 dorsally on LPLC2 dendrites leads to more synapses through interactions with Cy2 on T4, T5D neurons. By contrast, lower levels of beat6 ventrally correlate with fewer synapses. Altering this gradient by removing beat6 alters the tuning properties of these neurons along the dorsal ventral axis. At the other end of LPLC2 neurons in the brain, high levels of DPR13 correlate with more synapses onto the giant fiber. This gradient correlates with the strength of the escape response, with a more robust response to dorsal looming cues than to ventral ones. And by uniformly increasing the level of DPR13 across the LPLC2 population, we lost the graded response to looming stimuli at different positions along the dorsal ventral axis. Now, Mark has found synaptic gradients in all 20 VPN cell types he analyzed by EM. So this is a very common wiring strategy for visual motor transformations in the fly. And it'll be interesting to see whether this is a more general mechanism for sensory motor transformations in flies and in mammals. So in summary, the diversity of Ig superfamily proteins contributes to synaptic specificity in different ways. Matched expression of heterophilic molecules regulates synaptic partner matching. Vast molecular diversity with thousands of isoforms is necessary for normal development, in part through self-avoidance and controlling the appropriate composition of multiple contact synapses. Different proteins such as turtle are localized to and are required for synapse formation in specific subdomains. And finally, gradients of recognition molecules of this large family regulate gradients of synapses. Now, I have a lot of people to thank and acknowledge here. So there's part one and part two, because part one was really a review of lots of work that have been done in my lab over the years. And I don't expect you to read these names, but I really want to make 2 points. One is this work was done by a ton of people, and we had a ton of collaborators, really fabulous collaborators. And I have to say, one of the really great parts of this career has been the ability to interact with so many scientists and share in the fun of doing this science. So this is a fabulous group of people, and I feel incredibly fortunate to have had this experience. Now, the second part of the talk was a little more focused, but it also required lots of work from lots of people. It was driven by Mark Dombrowski, and Mark has been jointly mentored by me and Gwyneth Card. And I want to emphasize as well, of course, that much of what I talked about today is dependent upon extraordinary progress made in EM reconstructions, in work really pioneered and pushed at Genelia to give us a complete connectome. And there is much, much, much more to learn from the connectome and combining it with genetics and molecular tools to really understand principles of brain wiring. Thanks for your attention. I'd be happy to take questions. Oh my God, it's a non-believer in molecules. He's not a trainee. I think of him as a trainee.
Yeah. I'd be honored to be a trainee. No, please do.
I'm so sorry. Perhaps a naive question, but I'm wondering what's upstream of the LC. I'm sorry, I can't remember the acronym right now.
I'm impressed you just got an LC. LPLC2.
LPLC, yeah. What's upstream of them that is there anything activity-based or...
I'd love to tell you something about activity and the role of activity regulating this. We have no data on that. We know that there's activity. There's a lot of activity during development. We don't really know what that activity is doing. It's being studied in detail by Orkun Akin at UCLA. What's upstream are the T4, T5D neurons, but then there's also T4A, T4, another 6 neurons. There are other inputs. The connectome is ridiculously complex. There are many, many other neurons. I mean, you saw from the numbers. I mean, 726 cell types. So there's a ton we don't know. We know a little.
And a follow-up to that, is there anything temporal about
I'm sorry, let me just, I shouldn't be so negative.
Sure.
The T4, T5 cells have been studied in detail. They've been studied in detail functionally. They are directional selective neurons in the 4 cardinal directions. T4 is the on pathway. T5 is the off pathway, and it's been studied in great detail and by Axel Boris, Tom Clarendon, and others. And it's an incredibly rich system.
Gotcha. And I'm wondering, is there anything temporal about kind of the activation or development of the T4, T3, B, C?
Yeah, that's a fabulous question. We are woefully ignorant of the developmental dynamics. As we get out to more peripheral part of the visual system, we have a better idea of sort of developmental dynamics of individual cells, but we really haven't focused on that. I'm sure Mark and others will do that in their own labs.
All right, we'll resume.
Okay, come on, Larry.
I couldn't resist.
Of course, absolutely beautiful, and I love every single one of your molecules, at least as much as you. I'm as curious, and I want to be, and as careful as you are, trying to figure out how they actually operate all these beautiful roles during development. And so, coming to the second part of your talk, then I can guess my question already is, if I see this beautiful gradient of more branching and more synapses, the question arises, what comes first? Do you think that it is branching that is regulated by the interactions and synapses are a consequence of that gradient vicinity? Or do you think it's a primary role in synapse formation and the branches are a consequence of that?
Great questions. I mean, we have no mechanistic insight at that level because we haven't looked developmentally. The morphology is incredibly complex. So to be able to follow branches in different layers when things are really squished together is a real challenge. So I think it could be that it's playing a critical role in making synapses, and a failure to make synapses leads to retraction. That's one model. Alternatively, they may need that interaction in order to extend out into the layer. So that simple, two alternatives to discriminate between one another, but actually to do it, as you know, to do it at a level that's compelling is difficult, and I think it really needs live imaging, which everyone should hear Robin's talk tomorrow about the importance of doing live imaging, because that's what you need to do. You have to see it.
Yeah, I had a question.
You get two.
I can wait.
Oh.
Do I ask? Okay. So, maybe I'm going to start by being almost as emotional as Sam, saying that me too, I attended the first EMBO axon guidance meeting, where at a time where I was not even working on axon guidance or anything, just for the sake of it, I was doing my PhD trying to clone olfactory receptor genes from sperm cells. So really weird. And the first lunch at the meeting, I was sitting next to this guy who asked me, and he said something probably like, I'm Larry Zipursky. I had no idea who he was, of course, and I had no idea what I was doing there. And then, he asked me, so what are you doing? And so I just told him what I was doing. And he remained absolutely calm and, asked a number of very insightful questions about what I was doing, so that I was really feeling super smart that I was working on such a great project. Anyway, I hope I've learned, thanks to that, to be as nice as he is with trainees when we're having lunch once in a while. Anyway, that was just a little side note. I was trying to think quickly about what would be the molecular / cellular rate limiting step that somehow converts differential level of expression of the adhesion molecules with different level of number of synapses. So one of them is what Robin was alluding to. Now I was thinking about something else. What else is making which other neurons are making synapses with these postsynaptic partners? And could there be some type of competition? And that basically what these molecules here are doing is introducing a bias between different presynaptic partners onto the same target, or maybe it's not the case at all. But that's my question.
Yeah, it's a great question. To actually mean it. I mean, we all tend to say when somebody asks a question, great question. I know it's a really important question. So, we have two different phenomena here, right? We have one gradient where you have different inputs, whatever, 100 different axon terminals that express graded expression of a recognition molecule that are making synapses onto the dendrites of one cell. So there, I think I would think that competition is playing a role at some level of leading to different numbers of synapses. On the other end of the cell, we actually have a topographic map. So there you have let's just call it, for the most part, a matching of retinotopic position. And they have different levels. There are different levels of beat 6 on dendrites. And we imagine, we haven't looked, that there is the same level of the interacting proteins on T4, T5D. So that's now not a competition. So it's different numbers. So I suspect that, and maybe Elena's going to talk a little bit about this this afternoon, that it's a combination of affinity, levels of expression, and more generally than avidity. But I think this is a really neat place to study this problem. Yeah.
Sorry, we've got a trainee question at the back here.
Hi, I'm Ben from Julia Fave's lab. And I was wondering, do you think that the gradient is determined early in development, or do you think that it can be experience dependent, where the gradient can change and allow for possible rewiring and plasticity?
Yeah, so I'm going to take the second question first. We have no idea. Okay. The first question is really cool to think about, because the cell bodies are salt and pepper. They're not organized in a graded fashion, right? So they're sort of some salt and pepper pattern between the optic lobe and the central brain. And they extend dendrites out that uniformly cover the receptive field. So one interesting possibility is that there's some graded morphogen-like molecule that's influencing, that's signaling to those dendrites. And as a consequence of that signal, the cells are then expressing different levels of DPR of beat 6 and of DPR13. So I think where the gradient comes from, that's what I'm betting on, but it could be lineage. We don't really know. But I think it's a really, really interesting question. Thank you.
Larry, a question about you're the only one who can give a talk where in the introduction there's like 12 different stories, each of them equally interesting. So one thing you barely talked about is this new, as far as I know, I don't know any other example, class of synaptic specificity where different classes of neurotransmitter receptors can be differentially located on the dendrites of one neuron, right? And so that's Pietro's work. How do you envision, so basically you're proposing that each classes of those neurotransmitter receptors is associated with a cell adhesion molecule that then dictates synaptic specificity with a presynaptic class of axons? How do you envision the cell biology of even targeting those different macromolecules, the different portion of the arbor?
It's an incredibly cool phenomenon, and we simplified it in that diagram because in that layer you have 4 classes of polarized dendrites in the 4 cardinal directions, and then you have, and of course they all have the same order, and they're overlapping. So a neuron that makes synapses selectively in the proximal region is in contact with every other part of the proximal distal axis from other neurons, and somehow its selectivity is for that particular spatial domain. So I'm sure that didn't make much sense to all of you. It's really complicated, and I did a lousy job of explaining it. But it really was what led us to think that what's important here is where you put the recognition molecule. Okay, and the most obvious thing is it's tagging along with the receptors because they're being localized. So this was sort of the motivation for Piero to do the first tagging and then use that as a way of trying to get at this question. And part of it is, we've been really frustrated, continue to be frustrated in this mechanistically looking at the relationship between synaptic specificity and making a synapse. So there really hasn't been a ton of progress made in understanding how synapses are made in the CNS in flies. We have all these molecules that do something to discriminate between different cell types, but we haven't made the kind of progress that's been made in mammalian systems where you actually have all these synaptic adhesion molecules. So the hope was, well, maybe we could get lucky and maybe some of these synaptic adhesion molecules would be stuck onto receptors. And so that was part of the motivation, to try to get to close that gap between the two.
Larry, this is more of a general question, but so this is now painting a more and more complex picture of how subcellular localization can be different. There are more and more families, and then in the mammalian brain, these like IGSF and cadherin superfamilies have selectively expanded. There are more neurons, more synapses, and everything. So how do you think about these codes becoming more and more complex? How do you balance that with redundancy? And this is more pertinent to how we design experiments to study these molecular pathways, not so much questioning that there is complexity built into the system.
Well, you should never have left worms, although you had to deal with redundancy in worms, right?
That's what led me to that, right? I should never have worked on cadherins.
Right, cadherins and worms, right? I mean, should be simple. I mean, I think what we look for is first, just experimentally, we look for places where there is no redundancy so we can figure it out. But, as you saw in Hagar's talk yesterday, in the mushroom body neurons, the number of DPRs and their incredible temporal pattern of expression, I don't know what that means. And I've had trouble really articulating, I think, what the complexity is like. And so when I look at the EM, like the one I showed you, and the number of different processes, neurons have to make a ton of decisions, right? And I'm sure timing is important, but there's a lot that's happening at the same time. And it's very intriguing to think about, well, how does redundancy play into? Is it really redundancy, or is it overlapping function? Are they purely redundant? It's hard to imagine selection to keep things based on pure redundancy. So there's clearly this complex overlap. So I think from a practical point of view, we just go where there's as little redundancy as we can get away with, right? But I think the redundancy is a really important problem and a fascinating issue genetically, is how you deal with that.
Yumin?
Larry. Yeah, remarkable dissection of these codes. The first question looked like kind of partially answered already. So are these cues working at the same time, or do they have any kind of sequential program? And then the second question is that the outcome of these interactions, is that a resulting in sort of a specific connectivity, allow information to be channel specific, or whether it's a result of a diversity of synaptic functional states, or both?
I'm not sure I got the question. I mean, I think there's a lot there. So the issue of order, embarrassingly, we know very little about order. Again, ideally you want to have live imaging to look at order. We have a little bit of live imaging that's been done in the lab over the years. Robin's done a lovely job of that in more peripheral parts of the visual system. But combining the live imaging with the genetics, protein localization, you really should be able to figure out order of function and being able to look, when do you first see phenotypes? And I thought you were alluding to, in some part, that, of course, I've said nothing about signaling, right? I mean, signaling, and from Alex's trainee's talk yesterday on signaling and effect on microtubules and an actin, there's just, all of these are feeding into, all of them are signaling, even those that don't have transmembrane domains and cytoplasmic tails, they're all signaling in some way. And that, at least in the work that we've done, has remained largely a black box. I mean, it's ironically, that's how we got into the diversity, but we never came back to the signaling questions.
We have one last question at the back.
Yeah, hi, really amazing work. I was just wondering, in a bipartite question, in the interactions between LPLC2s and giant fibers, when you knock down DPR13 or overexpress it in giant fibers, is there change in the overall arbor of these two neurons, and that's why the overlap is kind of changed? And the second part is, when the change is there in the synapses between these two neurons, is the total number of synapses in any of these neurons changed, or is it remains constant but just goes to a different place with different neurons?
So, I don't know the answer to the question. I want to make the point that LPLC2 has well over 25 different partners, types, in that glomerulus. So the giant fiber is only one of them. And there are gradients on them as well. A simple answer to the question about morphology is the morphology on the giant fiber neuron, in a DIP-epsilon mutant, is largely normal. But I say largely, it doesn't look completely normal. And whether that's a consequence of not getting innervation or it's a consequence of another role. We know that DIPs and DPRs do play a role in cell survival in other contexts, so it's possible it's contributing, not in this case for survival, but some aspect of morphogenesis and maintenance of morphology.
We're going to take
a brief... I just want to make a very quick comment. Some very kind remarks that were made. I thought a lot about what I was going to say at the beginning of this talk, because I have a very emotional relationship with Cold Spring Harbor. My first meeting was here in 1978, and I could tell you, when I was a first-year graduate student, I can tell you about all of the different experiences I've had, and I just want to say I wish I were among the younger group. You have a lot to look forward to. This is a mecca. It's a mecca of molecular biology and cell biology, and an extraordinary place. So you're part of a fabulous tradition. Thank you.